Phenomics is the comprehensive study of an organism's physical and biochemical characteristics, connecting biology, data science, and engineering. The global phenomics market has grown rapidly, reaching $411 million in 2023 and projected to hit $1.2 billion by 2030. Phenomics helps address a critical gap in biological research by providing deeper insights into the relationship between genotype and phenotype. This transdisciplinary field is enabling new discoveries and advancements across various scientific domains. The image on the slide depicts a modern scientific laboratory setting with researchers analyzing data and using high-tech equipment, highlighting the technological and collaborative nature of phenomics research. The phenome is the complete set of physical and biochemical traits of an organism. It changes over time due to development, aging, or metamorphosis. The phenome includes an organism's morphology, physiology, and behavior. The concept of the phenome was first introduced by Davis in 1949 as the phenotype as a whole. It has been described as the phenotypic analog of the genome, representing the observable expression of genetic information. The slide provides a historical overview of the origins and development of the field of phenomics. It starts in 1949 when Davis first conceptualized the idea of the phenotype as a whole. In 1964, Soule described the phenome as the phenotypic analog of the genome. The term phenomics was coined in 1996 by Stephen A. Garan at UC Berkeley and Lawrence Berkeley National Laboratory. The field gained momentum in the early 2000s as genomic technologies advanced. The timeline and key milestones helped trace the evolution of this important scientific discipline. Genomics is the study of the complete genetic material, or DNA, focusing on sequencing, structure, and function of genes. Phenomics is the study of observable traits that result from the interaction of genetics and environment. It analyzes the physical and biochemical characteristics of organisms. Genomics and phenomics are complementary approaches for understanding biological systems. Phenomics helps bridge the gap between genotype, genetic makeup, and phenotype, physical expression. Together, these two fields provide a more comprehensive understanding of living organisms and how their genetic information is expressed. High-throughput phenotyping technologies allow us to rapidly capture detailed trait data across many samples using automated systems. These large-scale data collection efforts generate massive phenotypic datasets that require robust data management and organization. Analyzing these multi-dimensional trait datasets can reveal complex relationships and patterns that we can't see with traditional approaches. Integrating phenomic data with other omics data like genomics, proteomics, and metabolomics provides a more holistic understanding of biological systems. The slide introduces the concept of the genotype-phenotype relationship, which is central to understanding how an organism's genetic makeup interacts with its environment to produce observable traits and characteristics. The genotype represents the genetic makeup of an organism, the fundamental building blocks that determine its potential. The environment refers to the external conditions and factors that can influence the development and expression of an organism's traits. The interaction between genotype and environment is a crucial process, where the genetic information interacts with environmental stimuli to shape the final phenotype, the observable physical and behavioral characteristics of the organism. This interplay between genotype and environment is a dynamic and complex process, and understanding it is key to fields like genetics, evolutionary biology, and medicine. Measuring plant traits is critical for breeding and research, but we face challenges in making these assessments more quantitative and reliable. Standardizing protocols across research groups will help ensure consistency and enable better comparisons. The massive datasets we're generating in plant science require robust data management solutions to handle the petabyte-scale information. Developing advanced computational tools and software is key for analyzing the complex, multidimensional data we're collecting. Integrating diverse data sources across different scales, from molecular to whole plant, is a major challenge we need to address. High-throughput phenotyping is a powerful approach that combines automated image acquisition, machine learning algorithms, non-invasive monitoring, and real-time data acquisition. Automated image capture systems can collect thousands of images daily, providing a wealth of data on the organisms being studied. Machine learning algorithms are then used to extract meaningful traits and characteristics from this raw image data, enabling deeper insights. Non-invasive monitoring techniques allow for continuous data collection without disturbing the organisms, preserving the natural state. Real-time data acquisition ensures that dynamic traits can be captured and analyzed as they unfold, giving a comprehensive view of the organism's behavior and development. Together, these capabilities enable researchers to gather rich, high-quality data at an unprecedented scale, opening new avenues for discovery and understanding. Data science is transforming how we study and understand complex biological systems and organisms. Machine learning techniques like neural networks can identify subtle patterns in large, multidimensional phenotypic datasets. Computer vision algorithms can extract detailed quantitative features from images and video of organisms, providing rich data on their physical characteristics. Statistical modeling approaches allow us to analyze the relationships between multiple traits, revealing underlying biological mechanisms. Integrating phenotypic data with other biological information like genomics, transcriptomics, and metabolomics provides a more holistic understanding of living systems. Plant phenomics encompasses a range of environments, from field conditions to controlled greenhouses and automated platforms. These tools enable high-throughput screening for crop improvement and in-depth characterization of plant stress responses. Automated greenhouses with robotic systems can precisely measure plant growth and development. Drones and other precision agriculture technologies collect detailed phenotypic data from agricultural fields. Specialized imaging chambers allow for analysis of plant root architecture and other below-ground traits. Handheld devices can be used in the field to monitor plant health indicators like leaf chlorophyll content. Together, these phenomics approaches provide a comprehensive view of plant performance across diverse environments. The University of Arizona's Field Scanner is a large gantry system located in Maricopa, Arizona that moves sensors over field plots to continuously monitor crops in a desert setting. The Envirotron at Iowa State University is a specialized facility with interconnected growth chambers and a robotic rover that moves between the chambers to monitor plants in a controlled environment. The Donald Danforth Plant Science Center has advanced indoor and greenhouse facilities with multiple imaging modalities for comprehensive plant analysis, including automated conveyor belts and imaging stations. These state-of-the-art plant phenomics facilities allow researchers to gather detailed data on plant growth, development, and responses to various environmental conditions. Phenomics is the study of the complete set of observable characteristics or phenotypes of an organism, in this case microorganisms. This slide introduces 4 key areas of phenomics research in microorganisms: colony screening, high-throughput imaging to study growth patterns of microbial colonies, metabolic profiling, analysis of the metabolites produced by microbial communities or microbiomes, resistance phenotyping, characterization of how microbes respond to different antibiotics, environmental adaptation, studies of how microbes adapt to changing environmental conditions. Phenomics is the comprehensive study of an organism's observable traits and characteristics, known as its phenotype. In animal sciences, phenomics is enabling new approaches to behavioral analysis, livestock improvement, and wildlife conservation. Automated systems can track the movement patterns, social interactions, and responses to stimuli of model organisms like mice and zebrafish, providing detailed behavioral phenotypes. Non-invasive monitoring of growth, health, and production traits in livestock supports more efficient and sustainable breeding programs. Phenotypic analysis also helps monitor wildlife populations, assess habitat impacts, and guide conservation strategies for endangered species. Phenomics is a powerful tool for gaining deeper insights into animal biology and supporting advancements across the animal sciences. Phenomics is the study of the full set of observable characteristics or traits of an individual, their phenotype. This slide provides some key statistics about human phenomics research. Over 300,000 electronic health records are typically analyzed in phenome studies. Researchers track over 7,000 distinct measurable traits or phenotypic features. There are over 150 dedicated phenome research centers worldwide. The image shows a medical research facility with patients undergoing various measurements and tests as part of phenomic research. Phenomic data provides a comprehensive view of an individual's biology and health, which can lead to new insights and personalized interventions. Medical AI has a wide range of applications in healthcare, each with significant clinical impact. Disease classification is one of the most impactful, with an 85 score. AI can help diagnose conditions more accurately. Biomarker discovery, with a 78 score, allows us to identify new biological markers of disease. Treatment prediction, scoring 72, helps us determine the best therapies for individual patients. Patient stratification, at 68, enables us to group patients based on their unique characteristics and needs. PheWAS studies, scoring 65, leverage AI to uncover associations between genetic factors and disease phenotypes. These applications demonstrate the tremendous potential of medical AI to transform patient care and outcomes. Phenomics is the study of observable traits and characteristics, and how they relate to gene function. We'll explore 5 key approaches in functional genomics that leverage phenomics. Gene function annotation, linking genes to observable traits to build genome databases and model biological pathways. Reverse genetic screening, altering genes to observe resulting phenotypic changes, validating gene function and understanding development. CRISPR screens, systematic gene editing followed by detailed phenotyping, enabling drug target discovery and gene network mapping. Mutant characterization, in-depth analysis of altered phenotypes in model organisms and agricultural research. These phenomics techniques are powerful tools for understanding the relationship between genotype and phenotype, and advancing functional genomics research. Pharmaceutical research is critical for developing new drugs and treatments. I'll cover 4 key applications of this research. Drug target validation, confirming potential therapeutic targets through phenotypic studies. Compound screening, testing thousands of molecules to find those with desired effects. Toxicity profiling, identifying adverse responses to drug candidates. Pharmacogenomics, linking genetic variations to different drug response phenotypes. These applications leverage advanced laboratory automation, robotics, and data analytics to accelerate the drug discovery process. Metabolic engineering is a powerful approach for engineering microbes to produce valuable products like biofuels, natural compounds, and industrial enzymes. Engineered microbes can convert biomass feedstocks into biofuels through fermentation processes optimized using phenomics data. Modifying microorganisms can enable the production of high-value natural products, with phenotypic analysis ensuring consistent quality and quantity. Enhanced microbial strains can be used to manufacture specialized industrial enzymes, with phenomic testing confirming the desired activity and stability. These diverse applications demonstrate the versatility and impact of metabolic engineering techniques. Agricultural research is a critical field that aims to address key challenges in crop production and sustainability. One important area is crop improvement, where scientists identify desirable traits like enhanced yield and quality. Another focus is developing stress-resistant crops that can withstand harsh environmental conditions. Analyzing the various yield components, such as plant architecture and resource utilization, helps optimize productivity. Efficient use of resources like water and nutrients is crucial, as is preparing crops for the impacts of climate change. Overall, the goal of this research is to enhance food security and support sustainable agriculture. Phylogenetics is the study of evolutionary relationships between species based on their physical characteristics or traits. Comparative phenomics looks at how traits change across related species, revealing evolutionary adaptations. Evolutionary development examines how phenotypes, physical traits, change during embryonic development, tracing evolutionary pathways. Phenotypic plasticity explores how a single genotype can produce different phenotypes, offering insights into adaptation mechanisms. Trait-based phylogeny reconstructs evolutionary trees using phenotypic data, complementing genetic approaches. These phylogenetic applications provide valuable insights into the evolutionary history and adaptations of species. Phenomics is the study of the physical and biochemical characteristics of living organisms, especially plants. Modern phenomics relies on a range of advanced technologies to capture detailed data about plant traits. These include 3D imaging systems that can reconstruct the structure of plants, hyperspectral cameras that measure chemical composition, and automated platforms that collect measurements in the field. Thermal imaging cameras can detect heat patterns in plants, which can indicate stress levels. IoT sensors installed in agricultural settings also provide continuous monitoring data on plant conditions. Together, these diverse technologies allow us to build a comprehensive understanding of the complex traits of living organisms. Light sheet microscopy is a powerful technique for capturing 3D images of developing embryos and tissues with minimal photo damage, making it ideal for developmental biology studies. X-ray CT scanning allows us to non-destructively reveal the internal structures of samples, which is very useful for studying root systems, seed anatomy, and animal morphology. Remote sensing using satellites and drones enables field-scale crop phenotyping across large areas, providing valuable data for precision agriculture applications. These three imaging technologies, light sheet microscopy, X-ray CT scanning, and remote sensing, each offer unique capabilities that can advance research and applications in biology, agriculture, and beyond. Deep learning is a powerful technique for extracting meaningful features from complex biological images, like plant phenotypes. Pattern recognition algorithms can automate the identification of important phenotypic signatures, allowing us to rapidly analyze large datasets. Sophisticated data visualization tools are essential for representing the multidimensional relationships between different plant traits. Statistical modeling approaches help us understand the complex interactions between various phenotypic characteristics. Together, these computational methods provide us with powerful tools to gain deeper insights into plant biology and accelerate our research. This case study explores how we can use various plant phenotyping techniques to understand and measure drought tolerance in plants. First, we'll look at root phenotyping systems that allow us to image soil-grown roots and measure changes in root architecture in response to drought stress. Next, we'll discuss methods for measuring water use efficiency, tracking how much water plants consume under controlled conditions. Finally, we'll cover the use of thermal imaging to detect leaf temperature changes that indicate stomatal closure, a key drought response mechanism. Understanding these physiological responses to drought will help us develop more drought-tolerant crop varieties. This slide provides an overview of the key data sources used in human disease phenomics research. The largest portion, 45%, comes from electronic health records, which capture detailed clinical data on patients. Mobile health devices like fitness trackers and smartwatches contribute 22% of the data, providing continuous real-world health metrics. Genetic testing data makes up 15% of the inputs, allowing researchers to link genetic factors to disease phenotypes. Imaging data from medical scans accounts for 12% of the data sources. The remaining 6% comes from other clinical tests and measurements. Together, these diverse data sources give researchers a comprehensive view of human health and disease. Data privacy is a critical concern, especially when working with sensitive phenotypic information. We must ensure proper consent procedures and thorough anonymization techniques are in place. Reproducibility is a major challenge in our field. Developing common protocols and reporting standards will be essential to ensure the reliability and validity of our research findings. The digital divide creates uneven access to phenomics technologies, which can lead to disparities in research participation and the distribution of benefits. We need to consider ways to make these tools more widely available. There is an inherent tension between commercial applications and the need for open data sharing in public research. We'll have to navigate this carefully to balance proprietary interests with the broader scientific goals of our field. The future of plant and crop science is heading in exciting new directions, which I'll outline for you today. First, we're seeing a push towards multi-omics integration, combining genomics, proteomics, metabolomics, and other omics data to get a more comprehensive understanding of plant systems. Real-time phenotyping in natural environments is another key area, allowing us to continuously monitor plants as they grow in the field. Analyzing plant biology at multiple scales, from the cellular to the ecosystem level, is critical for connecting the dots between different biological processes. Democratizing these advanced technologies and making them accessible worldwide is crucial for advancing plant science globally. And finally, we're seeing the rise of AI-powered prediction models that can forecast phenotypic outcomes, unlocking new possibilities for precision agriculture. Phenomics is an exciting field that involves integrating diverse types of data to better understand living organisms. This slide highlights 3 key research opportunities in phenomics: computational methods, developing algorithms to integrate complex phenotypic data from multiple sources, understudied organisms, expanding phenomic approaches beyond model species to understand a wider range of life, climate adaptation, applying phenomics to develop systems that are more resilient to climate change. These areas offer great potential for innovative research that can lead to important discoveries and real-world applications. I'm happy to discuss these opportunities in more detail and how we might pursue them further. International networks, like the International Plant Phenotyping Network, facilitate collaboration and knowledge sharing among researchers worldwide. Specialized journals such as Plant Phenomics, Phenomics, and Journal of Experimental Botany publish the latest phenomics research. Universities offer courses and workshops on phenotyping methods and data analysis, and online resources provide training as well. These resources and training opportunities help advance the field of plant phenomics by enabling researchers to learn new techniques and share their findings globally. The future of phenomics is about bridging the gaps between genotype and phenotype, allowing us to better understand the complex connections between an organism's genes and its observable characteristics. Phenomics tackles multifaceted biological problems, addressing the challenges of understanding how genomes, environments, and developmental processes interact to produce complex phenotypes. Precision agriculture is an exciting application of phenomics, enabling data-driven farming systems that can optimize crop production and sustainability. In the medical field, phenomics holds great promise for precision medicine, where treatments can be tailored to the unique characteristics of individual patients. Achieving these goals will require global collaboration, building international research partnerships to share data, resources, and expertise across disciplines and borders.